think glycolysis is actually really easy. I don't think it's hard. I think the hard part, and it's not maybe hard for you anymore because you get a paper on your exam, is remembering these reactions, right? And obviously, understanding what's happening. So again, you have to understand what's happening. You don't necessarily need to, memorize everything, okay? So with that, I'm gonna ask you real quickly, I'm gonna write the whole mechanism down right now, discuss it, but while I'm doing that, take like 1 minute, if you have it with you, just open up glycolysis, okay? Like the PowerPoint, whatever it is, and look at those compounds, look at the enzymes, take 1 minute to memorize it, and then regurgitate it back on the paper and see how many you can get right. Because if you can do it, like, great. Okay, here's the thing. If you waste your paper writing down a mechanism for glycolysis where you could actually remember it, and maybe instead show the pushing reactions instead, your life would be easier. Does that make sense what I'm saying? Memorize the reaction and use your paper for actually something that's more important. Because glycolysis is easy to remember. But pushing those stuff and remembering how to use a Schiff-based mechanism, aldolase, enolase, right, mutase, isomerase, that's going to take more time, okay? So look at it right now, take 1 minute and 30 seconds, try to memorize it, whatever it is, and then write it down all on the paper. Does it get you? When we talk about glycolysis, and I think what I've kind of emphasized throughout everything I've said in all of my review sessions, all of these office hours, is that effectively you want to make ATP. You want to make energy because that's how your body lives, right? When we talk about glycolysis, okay, glycolysis in itself, the name, glycolysis, lysis means to split a glucose, right? Or not split a glucose, but to break something apart. Glycolysis is the splitting of glucose. That is what it is, okay? And maybe you understand this, maybe you don't, but my job here is to just briefly go over it, right? When you talk about glucose, how many carbons are in glucose?
6.
6, right? So, there's 6 carbons in glucose. Let me ask you this. How many carbons are in pyruvate? 3. And at the end of glycolysis, what's the product?
2 pyruvate.
2 pyruvate, right? And you just told me that pyruvate has 3 carbons, right? So what's happening here is that you're ending up with this 6 carbon splitting off into 2 pyruvate, each of them having 3 carbons, right? You're just splitting it. That's what I'm trying to show you. The idea that this thing 6 to 3 and 3, right? So the first half of glycolysis is what we call the energy investment phase. When we invest something, it's like investing our money, investing currency. Currency here is ATP, right? Currency is trying to make this reaction start, to make this glycolysis actually happen, right? So from steps 1 to 5, we call it energy investment. Now the next thing I'm going to say here, okay, is that when we talk about energy investment, then we obviously have to talk about energy harvestment. Harvestment is what? What's harvesting?
Cutting the end.
What's harvesting? What does it even mean? The definition of harvesting is to get something, to yield something, right? To get what you're trying to get, whatever it is. Crops. That's what I think of. Here, though, we're talking about ATP. We're talking about NADH. This energy harvestment, okay? We add in energy, invest, but then in the in the actual mechanism of glycolysis, we make ATP, right? And the ATP that's being made here is in step 7 and step 10, right here. Pyruvic kinase, phosphoglycerate kinase. So what's important to realize here is that each of these enzymes, okay, obviously it's telling you what you're doing, okay? So in an exam, which I'm trying to tell you, you want to memorize this, okay? Maybe on the exam you won't memorize the structures. Okay, write down the structures, but at least you're saving space for yourself. Like, you're gonna see what I'm talking about, okay? When you start out with glucose, the objective of steps 1 to 5 is to get this glucose, this molecule. You guys know what glucose looks like, right? Don't forget it. Right? I didn't put in the OHs, but you know where the OHs are. The objective of the first 5 steps is to take glucose and split it. We want to split it in half. But the important part here is that we want it to be symmetrical, or as symmetrical as it can get, right? So when we start with glucose, this is not symmetrical. I mean, I think that's pretty obvious. Like, there's literally an O right here. You can't split that in half. So what the first 5 steps are trying to do is eventually get to 2 glyceraldehyde 3 phosphates that look exactly the same, right? What you're trying to do is make this thing symmetrical so that eventually it looks something like this. Tell me that this literally looks symmetrical to you. It looks somewhat, right? Eventually you can split it up, okay? That's what this is doing. It's adding in ATP, right? It's using ATP, putting a phosphate on the 6th carbon. Does that make sense? It's a kinase. What is a kinase? What is a kinase? It's an enzyme. It adds phosphates. What's important though about a kinase is that it actually takes or transfers phosphates from high energy carriers. What are high energy carriers? ATP. Notice that kinase is where ATP is. PFK, which is kinase, ATP is there. When we talk about kinase, ATP is there. When we talk about kinase, ATP is there. Right? When we say kinase, we're transferring phosphates from high energy carriers. That's what a kinase is. And that will help you whenever you're trying to figure out what enzymes are doing, what's happening in a reaction. Okay? So we use a hexokinase, okay? And once we add a phosphate onto this, does anyone understand the objective of adding a phosphate onto glucose? Why add? I mean, I get it. At the end of the day, you're trying to split it up, right? But if we were really asking about the mechanism part of it, why do you add a phosphate to glucose?
To prevent fusion.
Say it again.
To prevent fusion through, like, I guess, to allow it, to prevent it from going outside the cell.
Yeah, exactly. When you take in glucose, you don't want it to leave. You want to trap it inside so you can continue doing glycolysis. That's what this is doing. Adding a phosphate is trapping it to do glycolysis. That's important. You should write it down because I saw it in your slides in terms of concepts you should understand. You should understand why we're adding phosphates. It's to trap it inside the cell to do glycolysis so you can keep bringing more glucose in and trapping it. Okay? So, we use hexokinase. We take a phosphate from ATP and we put it on glucose 6-phosphate. Yes, I didn't show ADP to PI. That's fine, doesn't matter. Now, obviously, you have the rest of these mechanisms. Fructose 6-phosphate, fructose 1,6, oops, forgot to put bisphosphate. Bisphosphate, okay? So what's happening here? When we talk about isomerases, she wrote in your PowerPoint, you should know what an isomerase is, you should know what an aldolase is, you should know what a mutase is. In all general purposes, and this is not scientific based at all, this is just for me to understand, and maybe it'll help you too. I don't know if it's scientifically correct, but it helps. When we talk about an isomerase, notice how it's a reversible step. What's happening here is that we're going from the glucose to the fructose form. So we're going from here to here, right? We're going from here to here. But in this case, there's only one phosphate right now. So pretend there's no phosphate here. So it's just moving from here to here and pretend there's a phosphate on there. I don't feel like drawing it at the moment. Okay? So it's just flipping. That's what the isomerase is doing. I mean, it's an isomer, right? You guys know what isomers are, right? So then when we hit on to right here, PFK1, phosphofructokinase 1, this is a really important step of glycolysis. I cannot begin to tell you that this is a main regulatory point. What I'm saying is that once you hit step 3, you are committed to glycolysis. There is no going back. You are ending up making 2-pyruvate, okay? So once we hit here, step 3, which is why I put a star here. I put a star here because you want to trap it. Step 1 is trapping it. You want to keep it in the cell. Step 3 is when we decide to commit to glycolysis, there's no going back. And that is why it's an irreversible reaction. It's irreversible. Now let me show you something, because these are all patterns here. There's nothing different from any of these steps. There's patterns. In every step with ATP, it is irreversible. Irreversible, 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 irreversible. ATP is going to be irreversible. I mean, you got energy. What's the objective of going back? There is none. You want energy, right? So, now after that, we have PFK1, phosphofructokinase 1. Has anybody gone over lecture 15, chapter 15 stuff yet, regulation? You have? If you haven't, that's fine. But if you have you gone through it? Okay. Can you tell me anything about the regulatory mechanism of PFK1?
I want to say PFK1, was that the phosphorylase that regulates it? I'm trying to remember. Like phosphorylase A and B?
So I think that has to do with glycogen phosphorylase.
Yes, you're right.
Yeah. No, you're okay. That's
I
okay.
remember the PFK1 having like standing out of my head, but I can't remember it.
It's okay. Don't worry. PFK1 I'm not going to go through because I'm going to save that for regulation on Tuesday. It makes no sense to do that. But PFK1 is very important for regulatory, hence the start. After that, if it's a kinase, guys, what's happening? You're taking a phosphate from ATP and putting another one there. You're putting it on carbon 1. That's what it's saying. You already have it on carbon 6, so if we're using a kinase, let's take the phosphate on ATP and put it on this to get fructose 1,6-bisphosphate. And that is what this is. Am I making sense right now? What's happening? Okay. Now here's where the aldolase mechanism comes in. Aldolase is very important. It's called a base shift mechanism. You should know the base shift mechanism. What's generally happening here is the carbon 3 and carbon 4. Carbon 3 and carbon 4. If I write this thing out, if I write down fructose 1,6-bisphosphate in the linear form, not in this form, but the linear form, okay? Just elongated. This thing is going to react with the base shift mechanism. It has to be linear, okay? Because ending up, right, I told you that you have to split this thing in half. You have to split this eventually in half. So what this thing does, and I can go through it later, the aldolase is going to actually cleave the linear form on carbon 3, and it's going to cleave on carbon 4. That's what's happening. That's the idea of the aldolase, to split this thing in half, and that is why you get dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. But there's a problem. I told you that typically we're trying to become symmetrical, right? But notice this thing is not symmetrical. Okay, and I believe this lower half might be reversed. Their OH might be here or it might be here. I can't really remember, but I can tell you that there's an OH on either carbon 3 and carbon 4. I'm sorry about that. But when you split this thing in half, you're getting 2 different compounds, right? Because they're not exactly symmetrical, but they're symmetrical enough, okay? So, in order to get what we want, right? Because you split it up and you get glyceraldehyde 3-phosphate, boom, I love that. That's perfect, so just check it off and you can go on to the next steps. But dihydroxyacetone phosphate, No. No. So what's going to happen here is that we're going to use another isomerase. Isomerase kind of rearranges this thing, and then we get glyceraldehyde 3-phosphate again. Make sense? Right? I mean, the main objective here is that you get glyceraldehyde 3-phosphate, 2 of them, right? You go through this whole process, okay? And you end up getting 2-pyruvate, right? What I'm trying to say here is that if you look at each of these enzymes, at least here, if you look at them, all of these enzymes are telling you exactly what's happening. They're telling you exactly what's happening. And that is why it is so easy to memorize glycolysis, okay, and keep your paper clear for other stuff. GAF3 dehydrogenase. After we make glyceraldehyde 3-phosphate, we're going to go ahead and do the second half of glycolysis. Steps 6 through 10, okay? Glyceraldehyde 3-phosphate dehydrogenase. That's the enzyme we're using here. This is step, I believe actually it might be step 1, step 2, step 3, step 4. Step 6. Yeah. Step 6 is a redox reaction. That's important to understand. It's a redox reaction. And I'm going to explain it to you. When we say dehydrogenase, what does that mean? What is a dehydrogenase? What does it do? Dehydrogenase, it takes off a hydrogen, right? So what's happening here is that glyceraldehyde 3-phosphate has a hydrogen on it somewhere. You'll see it in your slides. I'm not going to show you the structures because clearly it would make more sense for you to do that yourself, right? More practice, more understanding what's happening. I'm just showing you the basic portions of it. There's a hydrogen in this glyceraldehyde 3-phosphate. What's happening here is this enzyme, okay, is going to come in and it's going to do the reaction, the redox reaction. What does redox mean? Oxidation reduction. Usually whenever you see NADH, you automatically know it should be a redox reaction. So what I'm saying is that some part of the metabolite, this is a metabolite. Glyceraldehyde 3-phosphate is a metabolite. 1,3-bisphosphate glycerate, that is a metabolite, right? This is just an energy carrier, right? NADH is an energy carrier. So what's happening here is that glyceraldehyde 3-phosphate goes to 1,3-bisphosphate glycerate. If you are making NADH, which means that this should be NAD+, right? When you go to NAD+ to NADH, is that an oxidation or reduction reaction? Reduction, right? So what is that telling you about glyceraldehyde 3-phosphate to 1,3-bisphosphate glycerate? It's oxidized, right? But now notice something. In this reaction, if we want to say it's oxidized, right? So we know that there's a hydrogen that falls off of glyceraldehyde 3-phosphate, right? And we give it to NAD+ to make NADH. So once that hydrogen falls off, you have to realize it has to take place. Something else has to take place of it. So actually, a phosphate comes in. A phosphate gets added to the place the hydrogen was taken off. Does that make sense? So this phosphate is now going to be on carbon 1. Do you see what I just did? Glyceraldehyde 3-phosphate. There's a phosphate right now on carbon 3. As soon as I take off the hydrogen and I give it to NAD+ to make NADH, I need to fill up that spot that left the hydrogen. And I'm going to add a phosphate in, and I get 1,3-bisphosphate glycerate. This is really important. You need to add that phosphate. Why? Because in the second step, the phosphate that you just added is going to come off and it's going to make ATP. And I'm going to show you. Is everyone following along with me right now? Okay, does this make sense? Hello?
Yes.
Yeah.
Okay, so you understand step 7 or step 6, right? It's a redox reaction. Now, this one right here is a phosphorylation, substrate level phosphorylation. Doesn't really matter. You can look at that yourselves inside the slides. But what's happening here is that it's another kinase. A kinase, again, we're transferring phosphates off or onto high energy carriers. GTP, ATP, right? These are high energy carriers. So we're going to take a phosphate off of 1,3-BPG, and you're going to go ahead and put it onto ADP. I'm just showing you like this, and you make ATP. This is your first step that you make ATP. This is step 7. And it's done by phosphoglycerate kinase. Just so you know, by the way, in the end of all of this, what 3 steps are the most important steps of glycolysis? I haven't gone through it yet. Yeah, what's the 3 most important?
1, 3, and 10.
Yeah, 1, 3, and 10. Why? It's okay.
Very different.
They're like
Gluconeogenesis. Regulation.
Regulation? Yeah, it's for regulation. But also what I mentioned in the beginning, right? You want to trap it, okay? What did I say about this one? Okay.
The commit
Oh yeah,
step.
this is the one that gets committed, right? So you want to trap it, you want to commit it, and then the very last step is obviously to make pyruvate. It makes sense that the first and the last step are regulatory points. It's important to have these regulatory points. Why is it important? Why can't you have gluconeogenesis, which is the backwards reaction of glycolysis? Why can't both of those be the exact same? Why? Why not?
Because one not be regulated.
Yeah, they won't be regulated. It doesn't make sense. If gluconeogenesis has the exact same process going backwards to make glucose, it does not make sense and it is ineffective. The whole idea of the body, okay, is so that we're making energy, okay? If glycolysis makes energy and gluconeogenesis uses energy, does it make sense that as soon as I make this energy, automatically I'm going to go ahead and do gluconeogenesis? No, because that ATP could be used for our muscles, not just for gluconeogenesis. Does that make sense? And that is also why you cannot have 2 processes happening at the same time in the same cell. Do you think your body is going to do gluconeogenesis at the same time as glycolysis in the same exact cell? No. It's ineffective, it's inefficient. It doesn't work. Okay? Now I'm going to ask you another question. I know I'm just kind of like coming at you, but I want to make sure you stay awake too. So, if I say, what's the biggest difference between glycolysis and gluconeogenesis in terms of location? Where are they both happening? Yeah, just say it out loud, I don't care.
Muscle and liver.
Huh?
Muscle and liver.
Okay, so which one's happening in the liver, which one's happening in the muscle?
Glycolysis of muscle, gluconeogenesis of liver.
Okay, why?
Because you need glycolysis in your muscle to like move, and then the liver stores glucose.
Yep, okay, perfect. Now let me ask you another question. I'm just asking questions now. If I have hexokinase, okay, what's the difference then between hexokinase and glucokinase? What's the difference? Yeah.
Glucokinase is only for glucose and hexose for any 6-carbon sugar.
Is that true?
Probably not. I'm guessing.
Okay, because I didn't think that was the answer. I didn't know if you
I'm
read it
guessing.
somewhere that I didn't read it. So, actually, glucokinase, and you should write this down, actually. Glucokinase is an enzyme found in the liver, okay? You're not going to find this in any cell. If the cell is in the liver, you will find glucokinase. Does anyone understand why? Why do you find glucokinase in your liver but not any other cells? Because it's a very big process. You know that your liver has glycogen storages, right? Right? So I can go into there, pick out glucose whenever I want, right? Do you guys remember in your problem sets, GLUT2 versus GLUT1? And how GLUT1 worked when there was very little glucose, but GLUT2 had to be used when there was a lot of glucose? Does that make sense? Do you guys remember that at all? Yeah. Okay, because that's going to be on the next part of regulation. Glucokinase, okay, is found in the liver. GLUT2 transporter is also found in the liver. What it's saying is that this enzyme is only working. It is only working in the liver because this mechanism is only to take large amounts of glucose, not just any amount of glucose. It's large amounts of glucose. It's saying that if somebody eats a very high carbohydrate meal, okay, my blood glucose is now 15 millimolar, right? 15 millimolar is extremely high. So what's happening with that glucose transporter? GLUT2 is going to work effectively. It's going to pull up that glucose. And because it pulls up that glucose and it's in the liver, we use glucokinase to start putting it into either maybe gluconeogenesis for later so we can store it later. My whole main aspect here is that it's for the liver. Yeah, this is it. Mutase. A mutase, what it's saying is that you're just moving the phosphate that's on carbon 3 to carbon 2. That's what a mutase is. Will you get that on the exam? Maybe, I don't know, but it's definitely very easy to understand. When we talk about an enolase, what it's saying here is that to get from 2-phosphoglycerate and to get it into phosphoenolpyruvate, you have to make a double bond. When you make double bonds, something has to come out. Water comes out, okay, to make that double bond, to collapse it. Does that make sense? Okay? And then, of course, pyruvate kinase, you make ATP. Are we good? Yeah? Okay, cool. I just wanted to explain that because I understand that, look, you shouldn't waste your paper on this. No way. Okay, I'm going to erase it. Is that all right?
There's no way.
So mutase, in my opinion, mutase is more so moving something. It's not really, I guess you can say it's rearranging, but technically speaking, it's the exact same molecule. All I did was just move the phosphate from one place to another. When we talk about isomerase, we literally talk about something completely rearranging in a sense of that backbone might not be the same as what it was before. That's how I understood. Does that make sense?
What's with the 3?
This?
Yeah.
Oh, good question.
Oh, 3 carbon.
3 carbon. I forgot. Okay. I'm gonna erase it real quickly.
She did short
Was that helpful? Because I know that everyone kind of sees these stuff, but like these enzymes all have a job, and the job is literally written there for you. So it's not too complicated when you think about it. Does that make sense? Yeah? Okay.
Yeah.
Do you understand the fates of glycolysis in a sense of once I get pyruvate, where can I go with it?
Yeah.
Right? Okay, let me ask you. Can glycolysis be anaerobic or aerobic?
Both.
Yes, exactly. It can be both. If I go under an anaerobic pathway, what can I do? What can I do? Yeah, fermentation.
Or ethanol.
Okay. What is the first enzyme used for fermentation of alcohol?
Oh.
Fermentation to
Something
ethanol.
decarboxylate.
Yes. Pyruvate decarboxylase. What's the second step then?
Something dehydrogenase.
Or alcohol dehydrogenase, right? What the hell? I don't know how you know that so fast. Okay, now let me ask you this. Lactate, how do you get it? Yeah, lactate dehydrogenase. So, if you notice something, you have lactate dehydrogenase and you have alcohol dehydrogenase. In both of these fermentation steps, do you regenerate NAD plus or do you regenerate NADH? Why do you regenerate NAD plus?
For glycolysis.
Yes, to recycle it into glycolysis. Why? Why do you recycle it back into glycolysis?
To do glycolysis?
Yeah, to do glycolysis. If you don't recycle it, how are you going to do it? I mean, the whole point was that if you're at step 6 of glycolysis, right? You're making NADH. The only way to make NADH is to have NAD+. If you run out of NAD+, that's the same thing that we learned kind of in the last unit with free energy, right? You want to have a large amount of reactants to push the reaction forward, right? As soon as you run out of reactants, it doesn't really work out that way, okay? This is kind of, everything's conceptual. Like, as soon as you start thinking about it, like, it's not that hard. Okay, does everyone understand the Schiff-based mechanism? No? Well, I won't lie to you, it's not some mechanism that even I can make that easily. It's something that you'll have to redraw, like, over and over. Honestly, I can see something like that being on your exam. Drawing something and pushing it, 100% I can see that on your exam. Why? Because you already have a sheet with all this stuff on it. Makes sense. So, should I do aldolase? It seems like a lot to draw, but I can do it. Yes or no? Say it. Yes? Okay. So, when is aldolase used? When?
Step 4.
Step 4.
Step 4, right? What are you making? What is the reactants? What's the products of it?
Fructose 1,6-bisphosphate. The products is dehydroxyacetone phosphate and glyceraldehyde 3-phosphate.
Yes. So, yes. What's the objective of the aldolase? I told you this earlier.
To make it symmetrical.
To
Cleave it.
point.
To cleave it. Yeah,
never
to
mind.
cleave it. So, I'm not gonna write this all out at the moment, but fructose 1,6-bisphosphate. Can you imagine that in your head right now? I actually want to understand because I can imagine it. Can you imagine it? Can you imagine fructose 1,6-bisphosphate in your head? I'm a visual learner, by the way, so that's why. No, you can't. You can't. Okay, that's fine. We'll drop it. Oh, you. Oh, wow.
I think it's front and back.
Oh, by the way, apparently it actually is fully symmetrical. I remember though in one of my other classes it wasn't symmetrical. I'm not sure why. So you know how I told you that there's either an OH on this one or this one? I just checked your slides and it's actually on both of them. So better I tell you instead of me covering that up. Yeah, I made a mistake. Okay, this should be on both of them. So let's see. When I write PI, by the way, it's just another way for me to write, like, right, you get that, right? I just don't feel like writing it all, and that's like a little abbreviated notation. So when we talk about Schiff base is talking about this. It's this lysine, okay? There's a lysine, and it's like this little enzyme thing that looks like that, okay? And then, oops, I want to have it actually written like this. The lysine is on the inside, okay? This is an enzyme. It's like a little hole, okay? It's like the chymotrypsin mechanism that you saw in exam 1. It's this, the enzyme is here, okay? And the substrate is inside here. So what's happening here? This is the Schiff base right here. This is the Schiff base, okay? That's what we talk about, okay? So when you have this fructose 1,6-bisphosphate, the only way for the Schiff base mechanism to actually work is if this is in the linear form. So what we're gonna do is push it into the linear form. As soon as you push it in, do you guys have your computers? Because I don't feel like writing this all out. Yeah, no, I see it. I just want to make sure because it'll be very time consuming for me to write this all out. I'm not even joking with you. So what I will do is, if you have it pulled up open, like right now, I can walk you through the steps, okay? And at any point you don't understand, I'll go ahead and write it out. That is more effective, and it is not as time consuming. So we see the first mechanism, right? And I'm on the same slide as her where she has the whole thing written out. Not the individual parts because I find the individual parts a little bit more hard to understand. So we have this linear portion, okay? This is fructose 1,6-bisphosphate. What we have to realize is that if something is a base, usually bases are nucleophiles, okay? So what's going to happen is that this Schiff base is going to attack the electrophilic portion. What is the electrophilic portion on your computers? What is it? Carbon, the carbonyl, right? That's the electrophilic portion. You guys remember when I was telling you about how you always make a tetrahedral and then you collapse it? In these mechanisms, every single time a tetrahedral is going to be made and something is going to be falling off. Literally, I cannot even begin to tell you. Actually, fun fact, okay? When I was studying for the MCAT, they wanted us to know how to like draw certain organic molecules. I kid you not, every single thing was just making a tetrahedral intermediate and then just making it fall off. So when you see this, you should be seeing a tetrahedral intermediate, which is exactly what happens, right? The base is going to attack the electrophilic portion, right, the ketone portion, and then it's going to attach itself, meaning that as soon as I attach it, we've locked this thing inside the enzyme. That's what it's doing. It needs to grab it, it needs to bring it in so we can do the reaction. Does that make sense? Okay, so then the next part. It says that a base is going to come in and what? It's going to grab this thing. And what is it going to do? It's going to, what is it going to do? Well, y'all have your things, just read it.
Forms another double bond.
Yeah, so the base is going to come in, grab the hydrogen. It's going to create a protonated, obviously it's still protonated though. It's going to create a double bond with that, and then you get water. It grabs the water. Do you guys see what I'm talking about? Because I have to write it all out. You see where the water's coming in and stuff? Do you guys know from organic chemistry, leaving groups, right? And alcohol is not a good leaving group. It's not. So the objective here is that as soon as we grab this hydrogen here, okay, we grab this hydrogen, which is in your thing, it's going to collapse it. So we grab this, and then this thing is going to go here, right there, okay? Then what happens is the oxygen is going to go ahead and grab a hydrogen and it leaves us water. Does that make sense? You just need to have a good leaving group so that you can get, damn, this is right? Yeah. So you can eventually get this. Blah, 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 blah. You have water leave, okay, because water is a good leaving group. The whole point here is that you want this thing to become protonated. Schiff base, I don't know if she mentioned this in her video, but a Schiff base is really important, yes, because it cleaves, but it acts as an electron sink. When I say an electron sink, okay, I'm saying that if something is positive, like this, right? If something is positive, that means it needs more electrons, right? Because it's only at 4, right? Formal charge is 4, it should be at 5. It's not at 5 though. So this Schiff base actually acts as an electron sink, and what's going to happen here is that on carbon 3 and carbon 4, something here is going to get taken off, which is, I believe is the hydrogen, right? Hydrogen gets taken off. I'm sorry, this is really bad, but this acts as an electron sink. Electron sink meaning it needs more electrons. It's going to allow to do the cleavage part. This is where you want to get. You want to get to this protonated part, because what's going to happen here is that a base is going to come in and it's going to attack and grab this hydrogen. Obviously, this thing is going to fall here. It's going to create a double bond. Now, what do you think happens next? What do you think?
Attacks the cargo. Carbon 2. Attacks carbon 2. And then it goes to the next.
Right? What just happened? Carbon 3 and carbon 4 got cleaved, which is exactly what you're trying to do. You see how it acted as an electron sink, meaning it grabbed electrons? Does that make sense? Right? And all of a sudden, glyceraldehyde 3-phosphate is made, and this obviously this thing comes off and it looks like that, and that's gap 3, right? After that, you have this thing here. This thing here is going to be called dihydroxyacetone phosphate. Really, that last step, by the way, all it's saying is that you hydrolyze it. When you hydrolyze something, what does that mean? You add water to take it off of this whole, you want to take it off the enzyme now, right? You don't want it to continuously be on the Schiff, right? You don't need it there. So what's going to happen is that you add water, and as soon as you add water, this thing is going to eventually look like this. Let me make sure I draw it correctly. Right? And then that's DAP, right? That's what I call it. You see, all it did was that it added where the nitrogen was, it just added an oxygen. Obviously, there's more mechanisms to this, but that's what hydrolysis is, right? Or hydrolyzing something. You're just cutting it off from there. You're taking it off from this enzyme, okay, because you don't need it anymore. Are we good? That's a base shift. Does base shift make more sense to you now? What it is? Yes? Does it make more sense? Yeah? Okay, yeah? Does it make sense? Okay, cool. So yeah, that was base shift. I'm so sorry.
It's on your PowerPoint.
Yeah, it's on the PowerPoint. Yeah, it's on the last part.
It's slide 19.
So I went over that. Let's see. In her slides, she has the mechanism and then the step and then the very important parts of the step, right? Just so you know, the important parts of each step, you should definitely know, but there's a pattern. If something is reversible, so your delta G is somewhat kind of positive, you're always going to keep the products very, very low to continue the reaction forward, right? Because that's just in free energy type of stuff. The other thing, which I already told you, step 1, step 3, step 10, the very, very important parts of glycolysis. And that is why, because those parts are so important, when you go to gluconeogenesis, you'll see that those are the only 3 parts of gluconeogenesis where the enzyme changes, okay? And again, that has to do with regulatory steps, because if they were the same, it's ineffective, it's inefficient. So the next thing, I want to make sure. I think honestly I hit glycolysis, done. I think you understand glycolysis, yeah, right? Okay, so then part is gluconeogenesis. And this one shouldn't take that long, to be honest. Oh, but I mean, do you guys understand PPP? PPP, pentose phosphate pathway? Okay, there's actually a really quick way to do this, this is way easy. Okay,
Okay.
it's really easy to actually do this pathway. It's not hard at all. I think the only hard part about this PPP pathway, okay, is memorizing those, the structures. This is the only one that if I had to tell you, this is something that you should probably put on your little pathway sheet. You should probably write down the structures for this, okay? Because I think it's too much work to actually memorize those ones, okay? But there is a way to do it, I'm not gonna lie. So I'm not gonna really, I mean, whatever. Let me just think about this. Yeah, I'm gonna talk about that. Yeah. So what's the difference between a transalbilase and a transketolase? What is it? Does anyone know the difference between them? So what a transketolase does, is that it's actually transferring 2 carbons. That's all it does. It transfers 2 carbons. When we talk about transalbilase, we're actually transferring 3 carbons. That's it. You're just transferring 2 carbons, you're transferring 3 carbons. Do you guys have that one? Can you open up the one that looks kind of like this? I don't know if the thing shows up for you guys. It's like this thing. I'm sorry, I'm like trying to pull it up at the same time as talking. It's like this. Do you know that one slide that's like gray with the stuff on it? It's like a big pattern. Yeah, that one. What slide is that? 27. 27 is the best slide for the PPP pathway. I don't understand anything else, with all honesty. I think it's a little bit confusing, but that slide makes the most sense. First of all, in my opinion, what you have to understand about PPP is what's the point of it, where does it take place, and what exactly is happening. What are you making? What's the purpose of it, okay? The pentose phosphate pathway, the whole point here is that you're getting glucose, right? You're having glucose 6-phosphate. That is the entry point. Let's write it down. Glucose 6-phosphate is the entry point into the pentose phosphate pathway. So I'll abbreviate it as G6P. This is the entry point, okay? G6P is gonna come in. And I'm just gonna ask you, why is G6P going into PPP? Why? Why do you even go into the pentose phosphate pathway? Why? Why do you do it? So there's multiple reasons. Let's say, for example, you already have lots of energy being made. You already have lots of ATP, okay? It makes no sense to continue making ATP by glycolysis. Instead, we can go ahead and maybe we need to do nucleotide biosynthesis, or maybe we need to do fatty acid synthesis, okay? Maybe we'll do that instead of doing ATP. Maybe that's what we'll do. We don't need to do glycolysis, we'll go through it a different way. So G6P is going to use this enzyme called glucose 6-phosphate dehydrogenase. I'm just going to do it as this. This first part here is going to make, I believe, 6-phosphoglucolactonate. I'm just going to, what I said, okay? What I just said. Okay, I'm not going to write it down. Glucose 6-phosphate, no, it's 6-phosphoglucolactonate. So what's happening here is that you're getting NADPH. Okay, you're making NADPH. Now the next part here is you have to use a different enzyme. I don't think that enzyme is very important. I mean, I never really personally ever memorized it. I actually memorized this one because it's really important. This one, again, if you're writing this down on your note card, you'll have it already ready. This one here, I believe, is 6-phosphogluconate.
The enzyme?
Yeah, no, the actual compound.
Oh yeah, 6-phosphogluconate.
Okay.
Yeah.
6-phosphogluconate. Okay, whatever.
Yeah.
So you get this. In this step, I believe you have to add water into it, right? You add water into it, Maher?
Yeah.
Okay, you add water into it. So
CO2 leaves.
You're getting, yeah, CO2 is getting released, and you also make another NADPH. And you get this thing called ribulose 5-phosphate. Okay, so far what I'm telling you is the first main steps to get to ribulose 5-phosphate. As soon as you get to ribulose 5-phosphate, everything kind of gets confusing. You have sectulose 7-phosphate, you have xylulose 4-phosphate, and then you have, xylulose 5-phosphate, and then you have erythrose 4-phosphate. Those three compounds, what you should know about them is that they are involved in transketolase reactions, and they're involved in transaldolase reactions to eventually make glycolytic intermediates. That's a lot. I'm gonna show you, okay? And by the way, this image that I'm gonna show you right now is a very, very abbreviated version of it, okay? Because I don't want to write everything down for you. It's gonna waste time. So, I'm just gonna write right through the objectives of the pentose phosphate pathway. First of all, where does it happen? Where does pentose phosphate pathway happen?
Fatty tissue.
Fatty tissue, mammillary glands like adrenal cortex, right? These are fatty areas. They create cholesterol, they create steroids, they create hormones, okay? So we'll say that it happens in mammillary glands, adrenal cortex, right? Etc. Okay, so this is where it's happening. The objective is maybe we'll do nucleotide, I'll just abbreviate it as NT, nucleotide biosynthesis, right? What is nucleotide biosynthesis? Just making nucleotides, yeah. DNA, RNA, that type of stuff. Nucleotide biosynthesis. And then the other part here is you have NADPH. What do you think NADPH can be used for? Is it energy? Not really. It's actually for making fatty acids. It's for making sterols. It's for making cholesterol. Now think about it. This is happening in the mammillary glands, okay? Fatty tissue areas, basically. And then you're also making NADPH. And NADPH can be used to make fatty things like cholesterol. Do you see the connection here of the location and what it's making? Is it kind of clicking? Say yes or no. Yes. Okay, cool. So, fatty acid synthesis, cholesterol making, right? All of that. And this is from the NADPH. Okay, this is from that. This can make that, right? If you want to do nucleotide biosynthesis, that comes from ribose 5-phosphate. So notice, NADPH can go here and make fatty acid synthesis, or it can do cholesterols. And then we have ribose 5-phosphate, which will do nucleotide biosynthesis. So where's ribose 5-phosphate coming from? It's coming from right here. That's ribose 5-phosphate. So now that we have ribose 5-phosphate, if you don't have enough nucleotides, we'll go ahead and do nucleotide biosynthesis. But let's say that you do have a lot of nucleotides already. If you already have a lot of nucleotides, do you think you're going to continue making nucleotides? No. That is when we decide to do transketolase and we decide to do transaldolase reactions. Now, I wish I had a PowerPoint so I could actually show you what's happening. Because I believe I explained it to one group today during class, and I showed them where these carbons were coming from, and I think it made sense. So I guess what I'm trying to say is that, do you need help understanding where the three carbons are coming from? How you're moving them around? Yes? Okay, yes? I'm going to hold off on that for a second, okay? But, wow, sorry, let's do that, okay? So now you have ribose 5-phosphate, right? Ribose 5-phosphate can go into making erythrose 4-phosphate, it can go into sectulose 7-phosphate, and it can go into xylulose 5-phosphate. So I'm just going to show you, right? I'm going to say E4P, erythrose 4-phosphate. It can go ahead and make cellulose 7-phosphate, and then it can make xylulose 5-phosphate. So we can get these stuff. Now, okay, so what? You get all of this. So what? You get it. What do you do next? Well, I said that if you have already a lot of nucleotides, what you can do with these intermediates here, through transketolase and transalkylase reactions, you can actually go ahead and do, give me a second, I'm going to show you. So, on that image, on that paper that you, or the PowerPoint, what slide is it again, Maher?
27.
Slide 27, right? Of chapter 14. Sorry, chapter 14. Do you notice that the intermediates that are made are fructose 6-phosphate and the other intermediate made is glyceraldehyde 3-phosphate? When you do a transketolase reaction, once you get these right here, the objective here is to go and push it into glycolysis. You make glycolytic intermediates. Does that make sense? It's saying that if I already have nucleotides, okay, I'm going to go ahead and make these. I'm going to do it through transalkylase. I'll do it through a transketolase as well. And then I'm going to go ahead and make fructose 6-phosphate and glyceraldehyde 3-phosphate. Does that make sense? That's what's happening. That is exactly what's happening. And you said that you needed, oh, no, it's okay. Do you guys understand where the carbons, yeah, what happens?
You see those three things they like play into glycolysis?
So it's not like, oh yeah, erythrose 4-phosphate automatically just moves into it.
Yeah.
No, it has to do these reactions, but yeah, those are
Okay.
how they got, they're like the gateway into these stuff.
And that's glycolysis.
This is glycolysis, yeah. So, wait, I don't know her name. I'm sorry, what's your name? Yeah. Nikki? Oh, Michaela, I'm so sorry. Okay. Can you just tell me, do you need help moving the 2 carbons and the 3 carbons? Does that make sense, moving them? Yes or no?
Can you go through that? She was showing us, like, there were a couple of comments on our worksheet that were like following this carbon like through
Yeah.
the PPT process, and like she tried to show us, and I was like, I don't know what you're saying.
Okay. Who else needed help with that? Yeah? Okay. Okay, I'll show you in the end. I'll show you how the things move, so that makes sense. Does everyone else know how the 2 carbons move and the 3 carbons? Yes or no? Yeah? Okay, well, that's it. So I'll show you later, then. I'll show you later, whoever needs help. Okay, gluconeogenesis is really easy. I don't think I need to go through all of that. I think I'm just going to do one thing, though. And it's the Malate Shuttle, and it's the it's the very first step of gluconeogenesis. I'm just going to show you those 2, okay? And then we'll call it a day. Sorry, I don't know what time it is. Oh damn, it is 4:30. No, I got time. People be leaving, making me feel like I was going slow. Okay.